Hi everyone, my name is Sophia Willis, and I am a first-year master's student under the supervision of Dr. Gonzalez. I chose my graduate student presentation on milk fever in dairy cows because it is a huge problem in the dairy industry and it can be fatal if left untreated. Alright, let's go ahead and get into it. So first, what is milk fever? Well, the scientific term for it is hypocalcemia, and hypocalcemia is a condition where the blood calcium levels are far too low. So calcium is an essential mineral that plays vital roles in bone health, muscle function, and nerve signaling. While all breeds of cattle can develop milk fever, it is most common in dairy cows, especially Jerseys, because they are high milk producers, making them more susceptible because of the fall of blood calcium being greater than its replacement from the body. The onset of hypocalcemia in dairy cows is typically 1 to 3 days after calving, but it can also occur before calving as well. The development and growth of the calf and the production of colostrum utilizes very high amounts of calcium from the blood, which causes this deficiency. So, like I mentioned in the slide before, pregnancy is one of the main causes of milk fever in dairy cows. Such high amounts of calcium are drained from the cow throughout her pregnancy in order to maintain the health and development of her fetus. This being said, cows on their first or second calving are not very likely to develop hypocalcemia, while cows on their fifth calving or greater are at a much higher risk because over time they lose the ability to replace calcium quickly at the time of calving. And then I do want to note that feed management in the last 2 to 3 weeks of gestation is crucial because at this point the calf is growing at a very, very high rate. Finally, dietary calcium can also be too high and can cause a subsequent decrease in overall blood calcium when a supplement of calcium or something like that is too high for the cow. So calcium regulation and homeostasis is vital to a cow during gestation. I'm going to break down the mechanism of this process. So in order to prevent blood calcium from decreasing at the onset of lactation, the cow must replace extracellular calcium lost to milk. She does this by withdrawing calcium from bone and increasing the efficiency of absorption of dietary calcium. A dairy cow is programmed to go into the state of lactational osteoporosis, mobilizing bone calcium to help her achieve normocalcemia, which is just a normal amount of calcium, in early lactation. So this will typically result in a loss of 9 to 13% of her skeletal calcium in the first month of lactation. This is reversible in later lactations. So although this process might really stress out her bones, the main objective, which is to maintain normocalcemia, can be achieved by doing this. So it's pretty cool. So some clinical signs of the onset of hypocalcemia are anxiety and restlessness, which some examples of this are ear twitching and head bobbing, the cow being easily agitated or startled, muscle tremors, which can cause a stiff or uncoordinated gait, hyporexia or anorexia, weakness and inability to stand, and in some extreme cases, even the loss of consciousness. And then finally, decreased ruminal activity. All of you know just how important this last one is, as an impaired activity of the rumen can cause abdominal distension, inappetence, reduced body condition score, or even reduced body weight due to the typical microbial fermentation being minimized, causing this deficiency in energy provided for the cow. When it comes to diagnosis, it is important to always be on the lookout for these clinical signs, especially during the time periods in which a dairy cow is most at risk. And if hypocalcemia is even in question, a blood calcium test is the best way to properly diagnose milk fever. as some of these prevention methods are not guaranteed to prevent hypocalcemia in dairy cows. These prevention methods have been proven to decrease the chances of a milk fever diagnosis. Limiting stress might seem like an obvious prevention method, but there's actually a great deal of science behind it. Stress hormones like cortisol can disrupt the normal mechanisms that regulate calcium levels in the body. Stress can also impair the ability of cows to mobilize calcium from their bones and gut to meet the increased demands. Stress can also suppress the immune system, making cows more susceptible to infections, and then in those cases which can further worsen calcium imbalances. And then finally, stress can lead to decreased appetite and feed intake, potentially resulting in inadequate calcium consumption. And then these last two are especially important, and I will go over them on the next two slides. So, oral calcium. Calcium administered to the fresh cow may arguably be called a treatment rather than a preventative measure for hypocalcemia. Briefly, the concept behind oral supplementation is that a cow's ability to utilize active transport of calcium across intestinal cells is inadequate to help her maintain normal blood calcium concentrations. By dosing the animal with large amounts of very soluble calcium orally, it is possible to force calcium across the intestinal tract by means of passive diffusion between, not across, intestinal epithelial cells. Best results are obtained with doses of calcium between 50 to 125 grams of calcium per dosage, depending on the cow's body weight. So in this figure, calcium and phosphate absorption mechanisms in the small intestines are demonstrated. Both calcium and phosphate ions can be absorbed paracellularly through pores in the tight junction and transcellularly. So here in A, it's showing transcellular absorption of calcium, which is stimulated by 1,25-dihydroxyvitamin D, which is just an analog of vitamin D, which binds to vitamin D receptor in the nucleus and stimulates production of apical membrane calcium channel protein to let calcium into the cell, and then a calbindin protein, which complexes the free calcium ions and carries them across the cell. So a vitamin D-dependent plasma membrane calcium ATPase pump utilizes energy from ATP to move the calcium into the extracellular fluids. A vitamin D independent 3 sodium and calcium exchanger also exists in the basolateral membrane to move calcium across the basolateral membrane. In B, we have transcellular absorption of phosphate, which is also enhanced by 1,25-dihydroxyvitamin D, which stimulates production of sodium phosphate co-transporter in the apical membrane that uses either 2 or 3 sodium ions to co-transport an HPO4 anion. Phosphate diffuses across a cell and exits the basolateral membrane via facilitated diffusion through a phosphate channel. The sodium ions that were used to aid calcium and phosphate transport are pumped into the extracellular fluids by the electrogenic sodium and potassium ATPase pump in the basolateral membrane. So I know that was a lot, but that's kind of the breakdown of how oral calcium can help stimulate those calcium levels so quickly. So our next preventative measure that is especially important is the prevention of metabolic acidosis. So metabolic alkalosis is largely the result of a diet that supplies more cations, which are potassium, sodium, calcium, and magnesium, than anions, which are chloride, sulfate, and phosphate, to the blood. So in simplest terms, a disparity in electrical charge of bodily fluids occurs in animals fed these diets because a greater number of positively charged cations enter the blood than negatively charged ions. So this figure demonstrates the current hypothesis on parathyroid hormone, otherwise known as PTH, effects at the surface of target bone and kidney cells under various physiological conditions. So in panel A, we have under normal conditions, PTH is released in response to hypocalcemia, interacts with its receptor located on the surface of bone and kidney cells in a lock and key fashion. This stimulates G proteins and adenylate cyclase, resulting in production of cyclic AMP, which acts as a second messenger within the cytosol of target cells. This initiates mechanisms such as bone calcium resorption and renal production of 1,25-dihydroxyvitamin D to restore blood calcium concentration to normal levels. So in panel B, we have alkalytic conditions induced by high potassium diets induce a change in the shape of the PTH receptor protein so that it is less able to recognize and bind PTH, resulting in failure to activate the cell by producing cyclic AMP. And then finally, in panel C, we have magnesium is required for full function of the adenylate cyclase complex. Hypomagnesia reduces the ability of PTH-stimulated cells to produce cyclic AMP, resulting in failure to activate the cell. So I also know that was a lot, but that's kind of the breakdown of preventing this metabolic alkalosis. Two of the most common treatment options for hypocalcemia in dairy cattle are calcium therapy, which is simply calcium solution administered intravenously, typically through the jugular vein, and oral 1,25-dihydroxyvitamin D, which is what we've been talking about, supplements or feed additives. So the fastest way to restore normal plasma calcium concentration is to administer these IV injections of calcium salts, which most commonly is calcium borogluconate, and the amount of calcium that can be injected into a single subcutaneous site should be limited to around 1 to 1.5 grams of calcium. And then treatment with 1,25-dihydroxyvitamin D and its analogs can be a more effective and much safer way than using vitamin D, but problems are associated with timing of administration, and that turns into a big issue. So the problem of suppression of renal 1,25-dihydroxyvitamin D production can be minimized by slow withdrawal of exogenous hormone over a period of days after calving. And this is just an image of someone giving a cow with milk fever a sodium solution injection through the jugular vein. So I did some deep digging and found a lot of research on milk fever. There are fair amounts in general on milk fever in dairy cows, and I'm going to talk about one of the studies I found. So in this study by Braun and others, calcium and phosphorus supplements were used to treat 30 cows with diagnosed milk fever. 15 cows with milk fever were treated with 500 milliliters of 40% calcium borogluconate administered intravenously. This was treatment group A. 15 other cows with milk fever received the same treatment, supplemented with 500 milliliters of 10% sodium phosphate administered intravenously, along with 80 grams of calcium as calcium lactate and 70 grams inorganic phosphorus as sodium phosphate. And these were all administered orally via their drinking water. And this was treatment group B. So then the cows were monitored and blood samples were collected for 3 days to measure the concentrations of total and ionized calcium, inorganic phosphorus, and magnesium, and then the activity of creatine kinase. So for the findings on this study, we found that the two groups did not differ significantly with respect to the course of the disease. In each group, 14 cows were cured. A rapid and significant increase in serum calcium concentration from the hypo to hypercalcemic range occurred in both groups within 10 minutes of the start of treatment, followed by a slow and steady decrease to the hypocalcemic range. Calcium lactate did not prevent the calcium concentration from returning to the hypocalcemic range, and the calcium profiles of the two groups did not differ significantly. As expected, the treatment had very little effect on the concentration of inorganic phosphorus in Group A. And in Group B, treatment caused a rapid increase in the concentration of inorganic phosphorus to a maximum 20 minutes after the start of treatment. This was followed by a slow decrease in the phosphorus concentration to the normophosphatemic range. These findings confirmed that combined intravenous and oral administration of sodium phosphate in cows with hypocalcemia resulted in a rapid and sustained increase in serum phosphorus, but not in serum calcium concentration. So this modified therapy did not improve the success rate of milk fever treatment, and further studies are needed to improve treatment of milk fever. So, in conclusion, milk fever is a very common metabolic disorder in dairy cows caused by hypocalcemia during the final weeks of gestation or in the early stages of lactation of dairy cows. While various prevention methods, including managing stress and providing calcium supplements, can reduce the risk, ongoing research and treatment strategies such as intravenous calcium and phosphorus supplementation are crucial to improving recovery outcomes. And then finally, early detection and timely intervention remains a key in reducing the impact of milk fever on a dairy herd and their health and productivity. So that's all I have, and I just want to say thank you for your time, and I would be happy to answer any questions you have for me. Just shoot me an email. Thank you.